recent development which we made with an inorganic polymer, but it is basically a biomaterial. And it is polyphosphate. And the reason we are working at a University Medical Center, and the main emphasis of our work is always to find out which kind of enzymes are involved in the synthesis of inorganic polymers, like in our body, bone, or let's say in other animals with the silica skeleton. And so we were checking which enzymes are driving the biomineralization process. And we started with the most simple metazone organisms, the sponges. And now I cannot, also not this. Okay, this works. Okay, so we started with sponges, and we wanted to see if we can find any enzymes in the sponge skeleton, in silica, or also in calcareous sponges, in the enzymes there. And can we mimic these kind of reactions, this biomineralization process, by using enzymes? And we found in the silica skeleton of the sponges such an enzyme, and it was silicatein. But this was not so important for our biomedical interest, because we wanted to extend our work and want to work on bone. Until 2010, there was not a single enzyme known which is going to be involved in the synthesis of bone material. And then we discovered that initially, when we are looking in the deposits where we have mineralization, that we have not in our bone, it is not calcium phosphate, but we have calcium carbonate. And then we discovered that it is an enzyme responsible for this process, and this is the carbonic anhydrase number 9. And this is very crucial, that we have enzymes, and if we can modulate the activity of the enzymes, then we can modulate also the speed and the kinetics of the biomineralization. Okay, so we come back to the bone mineralization. Phase 1 is calcium carbonate deposition driven by the carbonic anhydrase 9. The product of this reaction is amorphous calcium carbonate, because only amorphous material in our body can be processed further and metabolized further. Then we ask the question how we can understand the transformation of amorphous calcium carbonate to amorphous calcium phosphate. And we ask the question, where is phosphate coming from? The organic phosphate was not enough, but we made such kind of a calculation, and we found that it is polyphosphate, a material which was neglected and which was not stressed very much previously. But then we found that polyphosphate is the substrate for bone mineralization, but we need to have an enzyme which is going to hydrolase the polyphosphate to the orthophosphate monomers. Then we got the amorphous calcium phosphate phase, and then finally the hydroxyapatite. Very famous scientists before, like Linus Pauling or Sylvie Kornberg, husband Arthur Kornberg, or Carl Lohmann, who discovered ATP, worked on this material, and they found out how the chemical structure of this polyphosphate is. And then if we look now more carefully to the structure of polyphosphates, we see one phosphate unit, another phosphate unit, which is linked together with a high energy anhydrate bond. Similar like it is in the ATP. Then, according to the findings of Kornberg and others, it was known that if we, or in the body, energy-rich bond is hydrolyzed, the energy which is stored in this bond is at least to a part transformed into a metabolically useful material into ATP. Then we checked further and we found that after degradation of polyphosphate with the alkaline phosphatase, by chopping off the terminal phosphate groups, we can determine ADP as a first step. So the alkaline phosphatase is releasing the energy which is stored in this high energy bond and is going to phosphorylate AMP to ADP, and then with the help of the underlying kinase, we get the synthesis of ATP. So we had the basis for the phosphate delivery of which we need for bone mineralization. Now, very recently, we published a paper in Chemical Reviews describing this kind of reaction, and this was really something new, that we have an inorganic precursor for metabolic energy, and described also the physicochemical basis on it. Polyphosphate is primarily formed in the megakaryocytes from where the platelets derive. And then in the platelets which are filled with polyphosphate, it is released, and it is released as a soluble 40-mer of polyphosphate. How is it made now, this polyphosphate? Of course, intracellularly. In mitochondria, we get the synthesis of ATP, and ATP is then released into the cytoplasmic space and is entering the acetylcalcisomes. And during that particular process, most likely polyphosphate is synthesized. Then this polyphosphate is released extracellularly and is functioning as a generator and as a substrate for the ATP synthesis in the extracellular space. So before this kind of discovery, nobody had an idea how extracellularly ATP is accumulating. And but after the discovery that it is coming from the polyphosphate, a lot of new developments became possible. But the question now is why we need at all extracellular metabolic energy. And then I'm always coming to the cartilage, and the cartilage is only filled with approximately 5% of the volume with cells, and in these cells we have ATP which is formed, but extracellularly in the cartilage we have only polyphosphate as a donor and not ATP as a donor for metabolic energy. And this I want to show here. If this was in Vietnam, it means soup is as a scaffold of the cartilage is not organized arbitrarily. It's highly organized into, it's not like this, but it's highly organized in a way that the collagen fibers together with the elastin is going to form the sheets around, for instance, the blood vessels. This is highly ATP and energy dependent, and this energy is coming from polyphosphate, which we discovered, let's say, about 6-7 years ago. Then we found that polyphosphate, if it is linked to magnesium and calcium, has a little bit different biological activity than, for instance, with strontium. I come to this, and finally it has also high activity, of course, against the COVID-19 virus. First, biomimetic encapsulation. We needed to fabricate a polyphosphate in the same way like cells are doing it, and we succeeded, and we fabricated nanoparticles with a diameter of approximately 100 nanometers, exactly like it is in the cells. It is amorphous and therefore prone to the enzymatic hydrolysis. This achievement was patented and granted worldwide. The issue now is, what can we do with this polyphosphate? So we were first thinking, can we apply it as a component in the bioink useful for 3D bioprinting? This is what we did, because our idea was in the future we want not to apply the cells in a high concentration, but we want to apply it at a lower concentration to go together with polyphosphate so that the cells can proliferate and differentiate. So we were basically more or less the first group who would really demonstrate also in these 3D printed tissue-like units that the cells are proliferating. So I cannot close this. Okay, and this is a growth kinetic of the mesenchymal stem cells, and after 5 days we get a peak, which is due to the activity of soluble polyphosphate, and after 20 days we get a second peak in this kind of 3D printed tissue units, which is due to the nanoparticles, which are then released and serve as a polyphosphate substrate. Application for biospinning, then application for blood vessel formation. We have now, together with another company, we have designed a machine which is able to make 3D printing vessels. And in these kind of vessels, we can implement and supplement and include polyphosphate so that the cells can proliferate and differentiate in these kind of printed vessels. Drug delivery, we can encapsulate in this polyphosphate because it is charged. We can encapsulate charged bioactive low molecular weight compounds like dexamethasone or ascorbate. When we add now this kind of polyphosphate in the bioink to the cells, and we see that in the bioink the cells differentiate and start to deposit hydroxyapatite minerals. It's like in our body. Publication bone regeneration, we included nanoparticles into microspheres, added it to the bone defects, and saw that here we use strontium polyphosphate. We saw that it is highly regenerative and is causing almost complete healing of the bone fracture in rats and in mice. Now, very recently, only 3-4 months ago, we found out that polyphosphate is an unusually powerful, regeneratively active material for wound healing, especially for chronic wounds, for instance, also wounds with a diabetic patient. And we have already performed some human studies, and we saw these kind of chronic wounds that very difficult, if at all, possible to heal. We found from that in the region where we have this kind of regeneration, we have a high accumulation of vessels. And now we finished a study which showed that chronic wounds, which were not possible to heal with any kind of approach, we were able, after 2.5 months, to get a complete healing of these wounds. So polyphosphate is an extremely regeneratively active material for wound healing, and it is really astonishing. We are very much glad about this development. Then we found something else. It has also anti-COVID-19 activity, because everywhere in our body where we have a defect, blood platelets which are filled with polyphosphate accumulate and release the polyphosphate and contribute to the regeneration speed of this tissue. So we make it short, we come to these pictures. If we come to the tip of the virus in the receptor-binding domain, the part where the virus is docking onto the cells, we have a so-called cationic groove. Here we have arginines and lysine amino acids clustered together in the same spacing as we know it from the polyphosphate. And so we could detect that we have an absolutely perfect matching of the polyphosphate to the cationic groove, and then by using an in vitro and later on by in vivo assay, we found that polyphosphate is extremely potent inhibitor of the binding of the tips of the receptor binding domain from the virus to the adenosine cellular receptor. At concentrations 0.1 microgram per milliliter, which are much lower than usually in a healthy person, the polyphosphate level is. So we have, and now we are going to make clinical trials, of course, in order to verify this kind of very hopeful strategy also for the human therapeutical invention of the COVID-19 disease. We are making a nose spray system, and then the polyphosphate is combining together with the aerosol, which are coming anyhow from the lung, and is going to initiate the healing of the epithelia of the blood and respiratory airways systems. So we have now a very good hope that we can, with polyphosphate, start also to formulate a drug which is going to help these kind of strong pandemics. With no spray, which the people in different large journals in Germany highlight. Financial report for our work came from the DFG, from the BMBF, from our ministry, but also from the ERC program, and I got an advanced grant from the ERC, followed by 3 additional ERC proof of concepts. Before that, I had a human frontier science program, which is also very difficult to get. I received it. And then these are some people which are working in our group and contributing to the results which I presented just now to you. Many thanks for your attention, and if you have questions, I would be happy to answer those. Thank you so much.